Hello, I'm Lucia Gambuza with Modern Retina, and I'm here with Dr. Charles Wykoff, Director of Research at Retina Consultants of Texas in Houston. Dr. Wykoff was a clinical investigator on pivotal phase 3 trials for ENCELTO, the first and only FDA-approved treatment for macular telangiectasia type 2. And last August, he performed the very first commercial ENCELTO procedure outside of a clinical trial. Today, we're talking about what a year of real-world data use has taught us about this therapy. Thanks for joining us today.
Great to be here with you.
It's safe to say that MacTel is underdiagnosed. Has the availability of an approved treatment changed how referring physicians and general ophthalmologists are screening and sending patients your way?
Sure, this is a big step forward for patients, right? I've been following MacTel patients as part of the MacTel project for probably greater than a decade at this point. And there were previously an underserved population, right? They had these progressive visual symptoms in many cases without any available therapy. So it's a big deal to have something to offer these patients, and discuss with them and see if that they'd be a good candidate. And I have been talking broadly with referring physicians and eye care professionals at large, as have many of my colleagues, because it's a big deal to have something now available for these patients that before did not have anything. So I do think there's growing awareness of this, right? MacTel is not a common disease in eye care, but I do agree with you that it's probably under-recognized in many areas. And so broad education about the findings in MacTel, the patient's symptoms related to MacTel, and the progressive nature of this disease in many patients is very important.
How are you identifying and selecting candidates for ENCELTO now? Are there patient profiles, disease stages, or imaging findings on OCT that you found are most predictive of who benefits most?
Sure. I think the phase 2 and phase 3 clinical trial enrollment criteria are a good place to start, right? So you first of all want to make sure the diagnosis is correct. And then on imaging, I do think it's important that there be the presence of ellipsoid zone loss. That was a key enrollment criteria in the clinical trials. Some of the other findings on imaging and exam, I really am looking for absence of active neovascularization, right? So one of the potential adverse events of this disease itself, with or without Incelto treatment, is the development of a neovascular complex and bleeding in the macula. And so I really don't want to have an active neovascular complex. I actually try to make sure patients are off of anti-VEGF agents and don't need additional anti-VEGF agents before I consider them a potential candidate in that eye. And then, patient conversation is also really important for eligibility criteria, who I think could be a good candidate. And in particular, I'm looking for patients that notice that they have a visual problem in the eye that we're talking about, and they also notice that it's getting worse. And I kind of want, I really require both of those things to think that if a patient is eligible, because what we don't want to enroll is patients that A, are asymptomatic, and B, patients that are not getting worse. And the reason for that is Incelto does not make patients better. And I am very clear with patients about that. We're not trying to improve their visual function. I certainly wish we could, and hopefully in the future we'll have therapeutics for MacTel that can improve their visual function, but that's not what we think Incelto does based on all the clinical trial data that we have. And so if patients aren't noticing worsening and they're stable, then I really kind of hesitate to move forward with surgical implantation. And many times in patients like that, I'll educate them about how to track their visual function, right, using an Amsler grid, or sometimes I'll actually perform detailed visual field analyses like microperimetry to follow them over time to just confirm stability if they think that they're not progressing. But many patients do feel like they're progressing, like they notice like that their metamorphopsia or their little areas of scotoma or difficulty reading is getting worse over time. So that's what I'm looking for. Patients that notice they have a problem and it is progressing over the last 6 to 12 months.
So, once you've identified the right patient, can you walk us through what the actual implantation procedure looks like?
Sure. And many patients are bilaterally symptomatic. And so we do have a discussion frequently about which eye, both eyes, which eye first, right? It's sort of a question. And my typical process there is that if a patient is interested in doing both eyes, I will often start with their worst eye. And patients often know which their better and worst eye is. If they don't, we walk through that sort of process together to help pick. And I pick the worst eye only because I say to patients, look, this is a new implant for you. No one's ever put it in you before. We sort of know the general general risk benefits and alternatives for patients, but you're unique. And let's make sure that if there is a problem, let's say there's a reaction to the implant or something that I think is extremely rare, let's start with the worst eye first. That's usually what I do in patients that want bilateral implantation. And then to your question, the surgical procedure, I think fits nicely within the wheelhouse of what retina specialists are used to doing. You actually really, in most cases, don't need a vitrectomy machine. So in the operating room, we sterilely prep the eye in standard fashion and then make a little conjunctival peritomy, usually 7 by 7 millimeters. I usually place it inferotemporally to keep the implant out of the visual axis. And then you make a 3 millimeter wide sclerotomy, about 3.75 millimeters posterior to the coronary scleral limbus. You implant the ENCELTO implant after placing a double-armed 9-0 polypropylene suture through the titanium fixation loop. And then essentially you use that polypropylene suture to fixate the titanium fixation loop just inside of the scleral wound. So the propylene suture is not a wound closure suture. It's just to attach the ENCELTO implant just inside of the scleral wound. And then the wound itself is closed with two 9-0 nylon sutures with knots that are buried. And then you make sure that the polypropylene suture lays flat, and then you close the overlying conjunctiva and Tenon's capsule. And importantly, you need to confirm the intraocular location of the implant before you finish the procedure. So again, all these steps are within the wheelhouse of a retina surgeon. I think the biggest challenge is manipulation of the double-armed 9-0 polypropylene suture. It's a fiddly suture, likes to kink, and so you just got to be very, very careful and cautious with it.
Okay, thank you for the details on the implantation procedure. You performed the very first commercial ENCELTO procedure outside of a clinical trial last August. Now that you and your team have had a full year of real-world experience, how has the procedure evolved in practice compared to what you observed in the clinical trial setting? Has anything surprised you?
It was a comprehensive development program. We had a really good phase 2 and 2 really good phase 3 trials to guide what we would expect in the real world. So everything I've seen has been consistent with that. I've implanted now approximately 20 of these commercially, and so far I have not had any reoperations for any reason. I've had a few patients with very mild inflammation, sort of in the post-operative routine period that goes away with a topical steroid taper. So nothing unexpected from my perspective. I think the biggest challenge we have with these implants is actually how to answer the question. The good question that patients have is it working? Right? Because patients want to know, is this thing working? Because we're so used to, as retina specialists, to give an anti-VEGF agent, for example, with an eye with macular edema for whatever cause, and the edema goes away. And so we can point to imaging and say, look, the drug is working. The challenge with ENCELTO is that the anatomy doesn't change. So there's no before and after difference on the OCT or on the fluorescein angiogram or the color fundus photograph. Like, I don't have anything definitive to look in there and tell a patient, yep, looks like it's working. And so really what we're going to have is natural history compared to the progression of EZ loss in these eyes over time. And so those analyses are just beginning now, right? Because we're only a year into FDA approval. And so I think it's really important to do those real world analyses where we look at the OCT imaging and we look at the ellipsoid zone loss before implantation and then after implantation, and compare that to what we would expect from the clinical trial data. And that will, I think, give us a better indication of sort of what is the real world efficacy of this, because that's the goal, is to slow the photoreceptor loss. And the last point I make is, patients, they want good anatomy, but what they really want is function, right? Patients want to see, and they want to see better over time than they would have without treatment. And so some of us are doing sort of more sophisticated visual function analyses, things like microperimetry over time, to try to follow these patients before and after implantation to see if we can document really what is the visual benefit for these patients. Because I think the phase 3 trial was really helpful there. It did show a meaningful reduction in the loss of microperimetry and also meaningful preservation of reading speed compared to the natural history in the control arm. And I think it's important that we try to validate that in the real world.
Where do you see ENCELTO, and you kind of hit on this already, encapsulated cell technology going next? Could this platform be used for other eye diseases beyond MacTel?
Yeah, it certainly could be a platform technology. It's fascinating technology where it's a little cylinder with these little string type placements inside of the cylinder where little cells are living on inside of these cylinders that then produce a therapeutic protein that then diffuses into the eye. It's really neat technology. It combines cell therapy with gene therapy. It's fascinating and remarkably well tolerated in the majority of these patients. The couple of the adverse events I do talk about is our meiosis and delayed dark adaptation. We know that from the phase 3 clinical trials. I do always discuss possibility of extrusion of the device and possible damage to other intraocular tissues, but I think all of those are infrequent and rare. It would be great, for example, to have an anti-VEGF agent that you could put in this and then place it in the eye and remove it if you needed to. And it could certainly be used for the delivery of other therapeutics.
Dr. Wykoff, thank you for walking us through all of this. I think our audience will find this really useful as they think through ENCELTO in their own practice.
Yeah, no problem. Enjoy the conversation.
And thank you to our audience for watching. Be sure to visit modernretina.com for the latest news and updates on MacTel and ENCELTO.